Sister?
Yes, Sophie, we will start. Let them join later.
Okay, sister. Okay. Good morning, everyone, and a very warm welcome to all connected virtually for the 5th day of the National Online Summer School on Biotechnology, sponsored by Department of Biotechnology, New Delhi, and organized by Marian Star Center, St. Mary's College, Autonomous, Thoothukudi. Faith is taking the first step even when we don't see the whole staircase. There is an explanation for everything. What matters is not the idea a man holds, but the depth of which he holds it. So let's begin today's session by invoking God's blessings through this prayer song.
We shall overcome, we shall overcome, we shall overcome someday. Oh, deep in my heart, I do believe we shall overcome someday. We shall overcome, we shall overcome, we shall overcome someday. Oh, deep in my heart, I do believe we shall overcome someday. We'll walk hand in hand, we'll walk hand in hand, we'll walk hand in hand someday. Oh, deep in my heart, I do believe we shall overcome someday. We'll walk hand in hand, we'll walk hand in hand, we'll walk hand in hand someday. Oh, deep in my heart, I do believe we shall overcome someday.
Now I invite Dr. A. Lakshmi, Assistant Professor of Chemistry, St. Mary's College, Autonomous, Thoothukudi, to introduce the resource person of today's session, Dr. R. Sri Priya, Assistant Professor of Zoology, St. Mary's College, Autonomous, Thoothukudi.
A very good morning to one and all connected here through this online platform, and a very warm welcome to the day 5 of the National Online Summer School on Biotechnology, organized by Marian Star Center, St. Mary's College, Thoothukudi, sponsored by Department of Biotechnology, New Delhi. It is my pleasant privilege to welcome the resource person of today's program, Dr. R. Sri Priya, Assistant Professor of Zoology, St. Mary's College, Autonomous, Thoothukudi. Ma'am did her UG at Lady Doe College, Madurai, PG in Integrated Biology at School of Biological Sciences, MK University, Madurai, and PhD in Biotechnology at School of Biotechnology, MK University, Madurai. To her academic credit, she has cleared her UGC CSIR JRF Net Fellowship in the year 2004. She has awarded DBT Rapid Grant for Young Investigators for the year 2013-2016. She has also awarded Marian Sundarlingam Merit Scholarship for the year 2002-2003 and Professor S. Krishnaswamy Endowment Prize for the year 2001-2003. She has worked as a research associate for the DBT RFG project and project faculty for the DBT IPLS program. She has more than 10 years of research and teaching experience. Under her guidance, 4 students have finished their PG project and 1 student has finished her doctoral degree. To her credit, she has written 2 book chapters and submitted 2 gene sequences. She has published more than 20 papers in national and international journals and proceedings. She has organized many conferences and workshops and acted as a resource person for more than 15 programs. We are fortunate enough to have such an eminent resource person amongst us today. Welcome you, ma'am. I also extend a very warm welcome to our Secretary, Reverend Sister Flora Mary, our Principal, Dr. Sister A.S.J. Lucia Rose, Deputy Principal, Dr. Sister Kulandithras, overall Coordinator of DBT Star College Scheme, Dr. Sister Arokya Genetius Alphonse, Dr. Sister Jessie Fernando, Head and Associate Professor of Physics. A very warm welcome to all the members of the organizing team. Finally, I welcome all the student participants of our college and other colleges. Once again, I welcome you all to the National Online Summer School on Biotechnology and wish you a very happy learning. Thank you.
Thank you, Dr. Lakshmi, for your warm words of welcome. Before moving into the session, I request all of you to turn on your camera for the photo session. Thank you. Thank you, dear participants. Now I invite the resource person, Dr. R. Sri Priya, to share her expertise on screening of recombinant organisms. Over to you.
Thank you, ma'am. Thank you for your warm words of welcome. And thank you, organizers, for giving me this wonderful opportunity to present this particular topic in this summer school. I'll share my screen. Is my screen visible?
Yes, ma'am. The screen is visible, ma'am.
Okay, I think I'm talking to a group of young and bright minds who are in their undergraduation in the different various departments of science. So maybe not related to biotechnology. So maybe in future they may get involved in biotechnology also. So my talk will be on a part of the basic biotechnology techniques. So, and the most important of all the techniques, the screening of recombinant organisms. So I think for people who are not from the life science background, it may be a bit difficult to follow this talk. So I think I have made my talk with more of lots of pictures so that you can get interested in the talk and you can understand what I'm going to talk about. Okay, so if at all you people have any doubts, you can just unmute your mic whenever you have something to talk, ask, and you can ask me the questions. If I'm going too fast, also you can say that, ma'am, we don't understand, and I'll come back to you. Okay, dear students, shall we go for the presentation? Okay, so this is first I would like, before going into why screening. First of all, I just want to tell you a basic thing about recombinant organisms. So I think you people with all the three lectures, what you had with the previous sessions, you should have a very basic knowledge about, am I audible, ma'am? Am I audible?
Yes,
Am I
ma'am.
clear?
Yes, yes,
Okay.
ma'am.
Okay, thank you. This I would just like to give you give you an overview of what are recombinant organisms, and then I will get into what is the importance of this topic what you are going to talk about today. Okay, first of all, I would like to tell you what are recombinant organisms. I think with all the knowledge from the previous talks, you would have a very basic idea about what are recombinant organisms. Okay, so recombinant organisms are nothing but organisms which have a foreign DNA put inside their genes. Okay. For example, you can take yourself. If you have a foreign DNA from some other organism, you can call yourself as a recombinant organism. So recombinant organisms have other names like genetically modified organisms, or you can also call them as transgenic organisms. So these are other names for recombinant organisms. So when you look into this, is just a small picture which shows you what is what is it and how it is. Okay, so this red piece, you can think it is a foreign DNA. Okay, or you can call it in the nomenclature of biology or biotechnology, you call it as a gene of interest. Okay, it is a foreign DNA, and you can get this from any source. You can get it from any living source. You can get it from a plant, you can get it from an animal, get it from a bacteria, or from a virus or a fungus, anything you want. So only thing is, this particular piece of DNA should code for something which is needed, which you want to have. Okay, and you can put it inside the host DNA. The host may be anything. So it can be, you can transfer this gene into a plant DNA, you can transfer it into animal DNA or a bacteria or a virus. And what the final thing, what you get is the host DNA with the foreign DNA, you call it as a recombinant DNA molecule. Okay, so this DNA can be just put into any organism, and that particular organism becomes a recombinant organism. For example, I just because you're all students, you get more interested with cartoons. I just show it with a, this is not a real thing, this is just a cartoon which I want which will make you get interested into this topic. So you can see, so this is a fish, and you can take a piece of DNA from a fish and you can put it inside the apple tree. Okay, you can put it inside an apple and you get what is called as a fishy apple. On the other way, you can take a piece of DNA from this apple and you can put it inside a fish, and you can call it as an apply fish. Okay, so so this is now possible just with the techniques what we have in biotechnology. So biotechnology had grown so much that you can take a piece of DNA from any organism and you can transfer it to any other organism. So that is what is the basic phenomena of biotechnology. So whatever lectures what you had was like when Dr. Jabbar Singh talked about how what are the vectors that carry the or molecules of DNA which carry the genes are called as vectors. And then Dr. Mahalakshmi told you how these recombinant molecules can be transferred into cells, because any DNA has to be transferred into cells. No, so similar. So that those are the technology. So they told you about the methodology of doing this. So I also would like to. So this is how this is done. So before going into the methodologies and things like that, I would just like to talk about a few genetically modified organisms. Then only you will get an idea. So what these are? Can anybody in this group can tell me what is a few examples of genetically modified organisms what you see in day-to-day life?
Ma'am, cotton and BT
BT
cotton.
Very good, very good. BT cotton.
Ma'am, golden rice.
Golden rice. Very good, very good.
BT
Okay.
brinjal.
So BT brinjal. Very good, very good. Nice. Okay, I have a few answers. I know, like so I'm talking to a group of people who have a prior knowledge about these genetically modified organisms. But for people who do not have, I just have to show you a few pictures of genetically modified organism. So this is this is a genetically modified fish, and the trademark name of this is a glowfish. Okay, so this particular fish carries genes from a jellyfish, and they can glow in the night. Okay, so think of having a aquarium at your home, and the night the fishes glow. So so this is a outcome of biotechnology, glowfishes. And this is a mouse which expresses the green fluorescent protein. Can you see the mouse which is in the middle is a normal mouse. The other two which are on both the sides are genetically modified. You can see the green fluorescence coming out through their eyes. Can you see them? Can you see them, students? Can you see them? Yeah, so these are mouse which are expressing a green fluorescent protein, which is from a jellyfish. Okay, and can you see this is a super mouse? There are two mice, one on the left, another on the right. One on the right carries the gene from human. The human growth hormone gene is transferred to them, and because of which the mouse looks bigger, right? It is a so it grows faster. Think if you can transfer this human growth gene hormone into a goat which can grow very fast, and which you can get the meat in just 2 weeks, whereas in a normal goat you have to wait for 3 months. Okay, so this is a product of biotechnology, and these are some of the examples from plants. So have you heard about blue rose? So blue rose, Ooty rose, you would have seen all orange, red, pink, white, yellow, but nobody would have seen a normal rose which is blue in color. So only a genetically modified rose can have a blue color, because rose plants do not have the capacity to synthesize the pigment blue color pigment, but you can transfer the blue color pigment from some other flowering plant, and you can transfer it into rose and make it a blue rose. So this was made by a Japanese company. Okay, so they have a patent over this particular technology. And I think you people will know what is, sorry for the spelling, is a golden rice. So rice, like rice is just carbohydrate, but golden rice, which looks yellow in color, carries the genes for synthesizing vitamin A, which is needed for your eyesight. Okay, so this is one of the important product of biotechnology, transgenic rice, which carries the vitamin A synthesis gene. And here you see a glow plant. Okay, so this is a plant which carries a gene from firefly, which are, like fireflies. So I'll be talking about later. Okay, and these are other examples: purple tomato and fragrant petunia. So per tomato, you can see they are all red in color. So what if you have a purple tomato which have a lot of antioxidant activity? And you also can have fragrant petunia. Petunia are those which these flowers are generally used in the bouquet, right? So they don't have a sweet smell, but you can take the gene from another plant which has a sweet smelling fragrant flowers, and take the gene and put it in petunia plant. You can get a fragrant petunia. Okay, so these are some of the, I just want you people to get into the talk so that you know what are genetically modified organism. Before I get into the topic, what is the importance of screening and all those things? So let me get into it. So this is an example of BT brinjal. So how do you go about creating a genetically modified organism? If I have to talk about screening, I should first talk about how you make a genetically modified organism. So when you talk about a genetically modified organism, so as the earlier talks, you had two talks which were this is a continuum of this talk is a continuum of the earlier three talks. So the first talk, when Dr. Jabbar Singh told that you need a vector to carry your gene of interest. So that is what it is. So here, can you see, so the upper picture, the human cell. So here, for example, you take a gene of growth hormone from the human cell and you put it in a vector. Okay, so this is a plasmid, the lower picture, which is blue in color. So this is a vector. So you can take the human growth hormone gene from the human cells. You can put it inside the vector, plasmid vector. So can you see, you can just cut the vector. That is what they say, restriction, endonucleases, digestion, and things like that. You just cut the plasmid vector. You can just paste the human growth hormone gene into these vector. And what you get is a recombinant DNA molecule, the blue and purple, what you see to the extreme right. Okay, so the blue and the pink, which carries, this is a plasmid, which carries a human growth hormone gene. So plasmid is a gene from a bacteria, so is a genetic fragment from a bacteria, and this purple is from a human genome. So this recombinant molecule has to be transferred into a cell, because these DNA cannot live outside a cell. No. So even though you do all these experiments outside, that is what, like Dr. Mahalakshmi told, you do it in vitro outside the cell, you have to place it back inside the cell, into the cell, by the way of like what Dr. Mahalakshmi told, transformation, transduction, and transfection. So any one of the methods, you just transform them into the human, into any cell. So the cell, so you call the cell into which you transform or you put back the recombinant molecule is called as the host cell. Okay, so for example, the host cell may be of any origin. If you want to put the gene into a plant, you have to take a plant cell. If you want to put the gene into a mouse, you have to take a mouse cell. If you have to put it inside a bacteria, you have to take a bacterial cell. So when I told, I didn't tell, so when if you had noticed me right, I wouldn't have told human or human or plant. I told only the human cell or plant cell. And the point here is, so whenever you do a recombinant DNA technology work, you have to do it at the cellular level. Like cells, or they are the basic components of any organism or any tissue. So generally, you cannot transfer a recombinant molecule right inside a human. That is, you can do it, but at a very higher level. But at the basic level, you have to do it only at the cellular level. So first, you transfer the recombinant molecule into a cell, and then you grow the cell into an organism. So that is the basics of doing it, creating a genetically modified organism. Okay, so now I am coming to the point. So like when you do it at the cellular level, you cannot just isolate one single cell, and you cannot just take the DNA, and it is not when you see in the diagram, the cell is so big, the DNA molecule so big, you think you can just cut open the cell and you can put it inside the cell. No, the cell, you whenever you talk about a cell, you can do the experiments. So when you come to the real experiments, you have to do only on it to a group of cell. It is not a single cell, right? So if you have to do a transformation or in a bacteria, you cannot do it in a single bacteria. You have to take a colony of bacteria which will have millions of these bacteria there. So then you have to transfer these plasmids, try to push in these plasmids into a group of cells. Okay, so when you do the experiment is not always successful. It always happens that if you do a transformation with 1,000, if I, for example, if I take 1,000 cells, only 50% of the cells, that is only 500 cells, may take the plasmid or the genital new DNA what you are inserting. Other 500 may not. Okay, so now please be with me. Like the transformation, what you call as a transformation efficiency is just 50%. So you cannot expect all the 1,000 cells to get the DNA. So it means that you have a ambiguity here. You have 500 cells which have the recombinant DNA, and you have 500 cells which do not have the DNA. So for example, if you are going to take the 1,000 plant cells and you're you are aiming to get a recombinant plant. Okay, so but you have done the experiment on 1,000 plant cells and only 500 of them have got the DNA. Then how about what do you think? Like don't you think it is a waste? Because you have to develop these cells into plants. You have to grow these cells into individual plants. That takes a very long time, and you have to grow these cells. It takes a large amount of resources like media, the culture room, and all those things. So when you do this, that you have to do what is at this point. You need this this part, what is called a screening. Okay, you have to screen for the recombinant or recombinant cells, that you have to find out which among the 1,000 cells have got the recombinant DNA and which do not have the DNA. Okay, or else you will be growing all the 1,000, and at the end of the day you will find only 500 of it have got the DNA. Sometimes your experiment will be an utter failure. No cell would have taken your DNA, and you will be growing no just the host cells and not the recombinant cells. Okay, so this is why you need the screening. So why do you want to screen the recombinant organism? What is the purpose? So as I told you, to select as you do the experiments only at the cellular level, you have to select that one cell with a foreign DNA from among millions of cells. I just told you for the namesake 1,000 cells. It should be, when you do an experiment, it will be million cells. Okay, so it will be a waste of time and energy to grow all the million cells, is it not? So you'll be am I right? Students, please unmute your mic and say if you have understood my point here. The methodologies, let me talk later, but I just want to know whether you understood the point.
Yes, ma'am.
Yes, ma'am.
Yes,
Am I
ma'am.
clear?
Yes,
Yeah,
ma'am.
okay, ma'am. Thank you, ma'am. Thank you. Okay, so you have to, from among the 1,000 cells, you have to screen out which are positive and which are negative. Okay, so then you have to confirm if the DNA is present there. Okay, if whatever the DNA what you have transferred into the cell, is it there? Is it there in the whether it has got into the DNA of the host cell? Okay, even if sometimes the DNA would get into the cell, because we are playing God here. Okay, do you know, like what we are doing is against the nature. It is not a natural thing. We are doing something against the nature. You are putting a foreign DNA into a cell, and you want that particular DNA to produce a protein what you want to do in a different place. Okay, for example, I am taking a human growth hormone and putting inside a mouse. So, and I asked it to make a growth hormone. Okay, how I should know whether this particular gene is working good in that new system. Okay, so I want to know whether the foreign DNA is expressing its character. Okay, whether the blue rose, when I put this blue gene into the rose, whether the rose is becoming blue in color. I have to first screen it. Then only I have to go to because these experiments, what I'm talking about, they are very time consuming. They take almost 3 years to 6 years to finish one particular gene transfer. Okay, so it is not a day's work. Don't think like just seeing the pictures. Don't get fascinated. Overnight you can do all these things. It takes almost 3 to 6 years. So end of 6 years, when you're not doing a screening and finally find out everything is negative, that will be a waste of time, right? Then you have to know whether the DNA is complete or is broken somewhere, because it may happen. Natural system, you just put in a DNA, you don't know, you cannot see, like you don't have any devices to see the DNA. You have to have some methods to see only. Okay, so that you don't know whether the DNA is full or it is broken. So for all these things, you need to have do a screening methodology. So here I'm going to talk about the different screening methodologies which is used to screen the recombinant clones. So the methodologies may be broadly classified into direct selection, and you can use reporter genes, and you can do DNA hybridizations. I'm going to talk about all the three sectors of screening of the recombinants. First, let me talk about the first method of selection, and the direct selection. I'm going to talk about antibiotic resistance. Okay, first let me come into this particular thing. Like antibiotic resistance is generally used as one of the methods to screen the screen recombinants. So let me be like, you may know what is antibiotic and resistance is going to, because I'm going to put a human growth hormone. What is this antibiotic doing here? So let me make it clear. So first, let me tell you the basics about the action of antibiotics. Generally, antibiotics kill bacteria. So this is a method of selecting bacterial cells. So leave alone the other cells like human cells or the plant cells. This is a method of selecting recombinant bacteria, because as Dr. Singaravelan yesterday explained very clearly, E. coli is used as a model organism for all these kind of experiments. Principally or basically, genes are transferred into Escherichia coli, and then only they are used for other purposes. So basically, you need to transfer genes into bacteria, which is a very important aspect in biotechnology. Okay, so for that, you can use this particular strategy, that is antibiotic resistance. So when you talk about antibiotic, doctors give antibiotics to cure your sore throat. So what does antibiotic do? It kills the bacteria which causes the sore throat. So the action of an antibiotic is to kill bacteria. A few examples, you may know it as ampicillin, what you eat today daily, the kanamycin, and there are so many other antibiotics. Okay, so what is it to do with the whatever I am talking about? Okay, so whenever you look into the vectors into which the genes of interest are cloned to be transferred into host cells, like for example, if I take a plasmid vector, all the plasmid vectors will have an antibiotic resistance gene in them. So antibiotic resistance means that you resist something. So when these plasmids code for proteins which can antagonize, or which can stop the activity of antibiotics, or they can degrade the antibiotics. So generally, whenever you design a vector, you always add an antibiotic resistance gene along with it. So whenever this particular plasmid is put inside a host cell, that is a bacterial cell, the bacterial cell will start having the capacity to grow on an antibiotic. That means generally, if E. coli, if I'll ask you a question. Like yesterday he told, like you have to plate the bacteria on Petri plates containing a media. So if I add ampicillin to this media and I put the bacteria, what will happen? Can anybody say? If I add antibiotic like ampicillin and I just plate the bacteria on it, will the bacteria grow? I need an answer.
ma'am.
They will not grow. They will die, right?
No,
So yeah, they will die. No, but when this particular bacteria has, when you transfer a plasmid containing the antibiotic resistance gene into the bacteria will start having the capacity to grow on a ampicillin containing media. For example, if you have an ampicillin resistance gene in the plasmid, and this plasmid is transferred into the bacteria can grow on the antibiotic containing medium. So why is it important here is, so whenever you transfer a target gene, can you see this is the blue color target gene. So whenever you put a target gene, that is your gene of interest, you also along with it send an antibiotic resistance gene into the cells. So every time you cannot, for example, when you do an experiment, you take 10 colonies of E. coli, and then you transform it with this plasmid carrying your target gene. Okay, so along with it, you will also have the antibiotic. This plasmid will also contain an antibiotic resistance gene. And after doing the experiment, when you plate it on a medium containing ampicillin, that's all the bacteria will grow. So when you do an experiment, I told you only 500 of bacteria will get the gene or the plasmid. The other 500 will not get. So when I plate it on a plate on the media containing ampicillin, that's all the cells will grow. Does all the cell grow?
No, ma'am.
So which cells will grow? Can anybody say?
Bacteria with target gene.
Which have resistance.
Very good. Only the bacteria which has the target gene carrying
Resistant
plasmid. The gene of interest carrying plasmid can only grow inside the bacteria. So that means that antibiotic resistance gene, although it is not anyway connected to your target gene, these are also introduced along with your gene of interest, so that whenever you do an experiment, you need not grow all the cells. So at the first point itself, you kill. So what happened to the other 500 cells which do not carry this plasmid? So what happened to those cells which do not carry the plasmid? They are all?
They will die.
They will die. Very cool. They will die because they do not have the antibiotic resistance. They all will be susceptible. They will be killed by ampicillin, which is present in the media. So it means that only those cells which carry the plasmid can grow on that media. So now what you are doing, you have screened. What you have screened only those bacterial cells which have your target gene. So this is the first method of selecting your recombinant plasmid. Am I right?
Yes, ma'am.
Okay. Okay. I think this is, so I will just tell you some of the common antibiotic resistance genes which are used on plasmids. That is kanamycin resistance gene, hygromycin resistance gene, ampicillin resistance gene. So I think whenever you people, you're talking about a plasmid, I think you will be talking in your classes, PBR 322, PUC 18. Whenever you see any plasmid, there you will see a small fragment of gene which codes for AMP-R. CAN-R. So it is nothing but a small fragment of DNA which carries the resistance gene in them, and these resistance gene also get into the cell along with your target gene, and they will make the cell to live on the antibiotic containing media. Okay. So next I'm going to get into a bit complex way of selecting the cells which have your target gene. So and this is a very important part, and this is a very well-known example of okay. So before that, this is a very nice pictorial representation. So this is a bacterial host cell, and this is the when you do a transformation, the plasmids get into the cells. Okay, so only when the plasmids, those cells which carry the plasmids will be recovered on an antibiotic containing media. Can you see? So if you have 5,000, if you have plated 1,000 cells, only you will get 500 cells which are growing. And when you take these cells, they will be all carrying the plasmid. So think of if you're not using this kind of selection, you just plate it on a plain media, all the cells will grow. So every time you have to pick up, so you have to do it, you have to do an analysis, you have to take the bacteria, you have to extract the plasmid. That is a very long procedure. So you will be playing around with host cells which do not carry the plasmid. Okay. Now coming to the next method of selection, that is blue-white selection. So this is a very important methodology in bacterial transformation of recombinants. Okay, So like this will be a bit confusing. I'll try to make it as simple as I can. Okay. So like how I told, every vector will have an antibiotic resistance gene. Some of the vector, so this is that is antibiotic resistance screening. This is another method of screening called as blue white screening. Okay. Okay, so to be very simple, let me take see, this is a small plasmid, and this plasmid contains a gene. So generally when they design the plasmid, they also include this particular fragment of DNA called as the LAC-C gene. Okay, so if you people would have gone studied about some operon hypothesis, I think you people even have it in your 12th, operon, lac operon. If you had studied about the lac operon, this LAC-C gene is a part of the lac operon which encodes for the enzyme beta galactouronidase. Beta galactouronidase enzyme. Okay, so what is the importance of this enzyme is, this enzyme which is encoded by the LAC-C gene, when it reacts with a substrate called as beta galactosidase. So the enzyme always reacts with the substrate. No. So this enzyme reacts with an enzyme called as beta galactosidase enzyme, and it will make a blue color precipitate. So this enzyme reacts. So this enzyme will react with beta galactouronide. Okay. So this reacts with the substrate to give a blue color precipitate. So why are we talking about it is that so there is a methodology where you can beautifully see the color change of colonies which have your gene of interest. Okay, so this particular plasmid, generally most of the plasmid vectors carry this LAC-C gene. Okay, so when you use these kind of plasmids for screening for recombinant technology, you take the plasmid, you put it inside the E. coli cells, and you plate the Escherichia coli cells on plates containing the substrate, that is X-gal. You call them as X-gal substrate, beta galactouronide. So this galactouronide substrate is also added in the plates. So when these cells carrying the LAC-C gene plasmid, when they grow on the plate, they will produce a blue color colony, beautiful blue color colonies. Okay, so they make a blue color colony. Okay, so but this is not enough for our molecular biologists. They have devised a very beautiful strategy to find out whether your target DNA is inserted inside the plasmid. Actually, generally, whenever I told you, no, whenever you insert a piece of DNA into a plasmid, you have to first cut the plasmid. You have to take the foreign DNA and paste the foreign DNA into the vector. But that pasting itself is not 100%. Okay, the transformation is only 50%. I told you the plasmid getting inside the cell itself is a 50% probability. The similar manner, a new fragment of DNA getting pasted in the plasmid itself is also another 50% probability. So sometimes what will happen is, when you just cut open the plasmid and you try to put the new gene in it, what will happen is the plasmid will once again, they may not take up the new gene. They once again can hold hands together and they recircularize without taking the gene of interest into them. So what finally you will get is, you cannot see all these events because they are done in only microliters of solutions in small plastic tubes called as micro tubes. Okay, so they are very small and you do it in a very small volume. All this reaction is done in 10 microliter of solution. So in that condition, you cannot expect see all these events. So whether the DNA had pasted in the plasmid, whether the inside the plasmid, whether the plasmid had gone into the cell, that you cannot see. So you can only use such strategies to find out these things. So this is one of the strategies used to find out whether the insert DNA had got into the plasmid. Okay, so so as I told you, when this plasmid, which contains a LAC-C gene, is put inside E. coli, they will produce a blue color colony. So this plasmid may contain your gene of interest or may not contain, because so I told you the ligation, that is the pasting of the DNA or DNA getting inserted, is 50%. So even though they have not got it, the empty plasmid also can sometimes get into it, and they can produce a blue color colony because there is a laxi gene. So what scientists have devised is they have made design such that you can insert your gene of your interest or the foreign gene right in the middle of the laxi gene, right in the middle of the laxi gene. So it means like if this is the gene, this is the yellow part where the gene of interest can be inserted. So what will happen if a piece of fragment, unrelated fragment, gets inside the laxi gene? The gene will be broken, and it will not be able to produce the enzyme beta galactosidase enzyme. It will not be able to produce, because of which, so when such plasmid which carries your gene of interest, when it gets inside the E. coli, and when you plate these, what kind of cells, what color of colonies you will get? What color you will get? What will be the color of the colonies?
Colorless.
It will be colorless. Very good. Beautiful. So it means that when you get a blue colony, it means the plasmid is there, but your gene of interest is not there in the plasmid. And only, so this is a very nice picture which shows you like this is the plasmid vector and this is the foreign DNA. Okay. So sometimes, so you can see that the first picture. Is my slide moving? Is my slide moving?
Yes, ma'am.
Yeah. So can you see in this
Yes,
first panel? So there is the blue, light blue is the laxi gene, and you can see that your foreign gene had got inserted inside the laxi gene. It means that what color of colony it will make, because laxi fragment is what you say inactivated. It means it cannot produce beta galactosidase enzyme. So when you plate these kind of colonies on the X-gal plate, what you will get is a white color colony. Okay, so colonies will grow because they have the antibiotic resistance in where some other part, but now because they are laxi negative, they will be white in color. But see the last, don't see the middle one, see the last one. When there is no insert, when your foreign DNA is not inserted, there is no red color here, no. So those, although the plasmid is there, even though the plasmid is there, it doesn't have the foreign gene. It means what color colony it will make? What color colony it will make?
Blue.
Blue color. So that is the basic behind a blue-white selection. That means even though a plasmid had been transformed into an E. coli, it may be empty also. So this is the next step experiment where you confirm whether the plasmid has got your gene of interest, because that experiment is very difficult. You may think like just cutting up a plasmid, putting your gene inside is very difficult. So there may be failures in the experiment. So this is a step to screen out whether your experiment had worked out properly, the first step itself. So you may have a plasmid there, but the plasmid may be empty. So you may have so many white, if you don't use this blue-white selection, you may have so many white colonies on your plates, but none of them will carry your gene. Although they have the plasmid, they may not have your gene of interest. So only those, so this screening will beautifully tell you whether there is your gene of interest present in the plasmid. So this is a real plate which shows, which has a mixture of colonies which are blue and white in color. So the plate contains the substrate X-gal, and also it will have the antibiotic. Okay, because it carries the antibiotic resistance gene, which is in the plasmid, will make the E. coli to grow. But only when there is a target gene in the plasmid, the colonies will look what color? Will look
Blue.
Hey, when there is a target gene inside, what color it will be? When you see
White.
the blue color, don't say
White.
that. Blue color, Ravi. When you see the blue color, don't say it is the right colony. What you're looking for is the white colony.
White.
White.
Inactivated. So laxi is inactivated because your target gene is there. When your target gene is there, it will be white in color. When the target gene is not there, it will be blue in color. So this is what is blue-white selection. Okay, fine. So shall we move on to the next methodology? Am I clear? If you have any doubts, we'll discuss it at the end of the session. Okay. So
Ma'am,
now-
can you explain that one more time?
Yeah, right. Okay, thank you. Because I know it's a bit confusing. This is a methodology to screen whether your ligation, that is your insertion of DNA into the plasmid had happened. Okay, so this is a screening methodology to find out whether the target DNA had been pasted to your plasmid, because every time before you do a recombinant organism, you have to make a vector which carries your gene, right? So this vector has to be prepared. When you prepare a vector, you have to first cut open the vector. So I will show you the earlier picture. First, you have to cut open the vector, and then you have to insert your DNA there. Can you see this picture?
Yes, ma'am.
Okay, right. You have to cut. So we are going to confirm. So in this experiment, it may also happen that this vector can religate, or it can recircularize. So it need not carry the gene. There may be a probability that this will once again close without taking up the new DNA. That may also happen. Okay, so we are going to screen whether there is this presence of this new gene, or there is no, only the vector had a vector is there, or the host vector is there. For this, we are going to do this blue-white selection. Okay, so for this, most of the vector doing this is done only with this can be done only with those vectors which carry the LAGZ gene. Okay, LAGZ is a part of LAC operon, which encodes for the enzyme beta galactosidase. This beta galactosidase enzyme, when it reacts with the substrate X-gal or beta galactouronide substrate, it gives a blue precipitate. It gives a blue color. Okay. So whenever you do this experiment in the plate, along with the antibiotics, you also add the X-gal to the media in the plate when you're going to do this experiment. So you take the vector, and then you think that you're doing this experiment, you're cutting this vector, and the scientists have designed this vector in such a way that the cut is made right in between the LAC gene. The cut is made right in between the LAC gene. When the LAC gene is intact or unaltered, it will happily produce this beta galactosidase enzyme. But once when your gene is inserted in between the LAC gene, it becomes inactivated, because of which it will not be able to produce the beta galactosidase gene. Okay. So when there is an intact LAC gene, there will be blue colonies formed on the media. But when your gene of interest is inserted and the LAC gene is disrupted or broken, no beta galactosidase enzyme will be produced, because of which your plates will carry only the will show white colonies. So the plates where you have the white colonies on the plates are those which have the plasmid which carries your target gene, and the blue ones are those cells which carry only the vector. Right? Am I clear?
Yes, ma'am.
Okay, thank you, ma. Okay, now let us come into the next very interesting another other gene. So this blue-white selection can be done only in bacterial cells. Okay, so you just have it in mind. They can be done only with E. coli bacterial cells, not with other cells. Okay, please mute your mics. You are disturbing the other people. Okay, so now coming to the next group of genes which are used for screening, they are called as reporter genes. Okay, so reporters, you mean like they go and report. So these reporter gene is nothing but a gene which can be, whose protein can be easily assayable. It means measured. Either they can be measured, or some biochemical test can be done on these genes and find out whether these genes are present. So similar manner, how antibiotic resistance genes are present in the vectors, some of the vectors also carry these reporter genes. Okay, reporter genes are very easy screening method things, because when you use these vectors which carry the reporter genes, when these vectors are taken into the organisms, you can see the so you cannot say, because whenever why I'm not talking about looking into your gene of interest. So your gene of interest may produce a protein which may not be screenable, or it is not visualized. For example, if I'm cloning an insulin gene from humans into a bacteria, how will I see whether the bacteria is producing insulin or not? Okay, so insulin cannot be seen with your eyes. But instead, if I put along with this gene of interest, if I add what is called as a reporter gene, whose presence can be seen, visualized, that will be good, no? So that will show that the vector had got inside. So that kind of thing you can have. So such genes are called as reporter genes. So generally, these reporter genes are present in the vectors along with your genes of interest, and when so this presence of these visual markers will tell you that your target gene is also present. For example, so this visually or assayable protein will produce gene will produce a protein which can be seen with your eyes. So let me take few examples. The first one is the luciferase gene, which is generally used for this recombinant, screening for recombinants. Okay, so the every vector which is used for many of the vectors which is used for animal transformations and plant transformation carry the reporter genes. So maybe so for higher organism, reporter genes are must. Okay, because you start the experiment with a group of cells, and end of the day, when you get a non-transformed plant and animal, that will be a big disappointment. So generally, you use this kind of visual markers, which without even doing a big experiment, you can just visually see. Okay, one such gene is called as the luciferase gene. So luciferase, I think people would have know what is this. What is this in the first picture? Can you identify this?
Firefly.
Yeah, firefly. So everybody would have seen this. So now those days we people in the olden days we used to play with this. Nowadays these are not present very frequently. Okay, these fireflies in the night they glow. Okay, you know why these are glowing? And their property, they need this glowing to attract their mates. They glow to attract their mates. And the reason why these are glowing is because they carry a gene called as luciferase gene in them. Okay, so this gene is a very lucrative gene which can be used for many experiments. So scientists now can take a DNA from any organism. So they took pain to take this luciferase gene from the firefly, and they have put it in the transformation vectors. Okay, this luciferase gene will produce this luciferase gene will produce a glow when a substrate called as luciferin is added to it. So what they do is this gene from the firefly is taken and it is put inside the vector along with your gene of interest, and this vector is transferred into plants and animals. So this is a reporter gene for generally used for plants. Can you see this is a tobacco plant which has got this luciferase gene, which has got the luciferase gene. Can you see the glowing tobacco? This is a tobacco plant which glows. When you now this generally these kind of plants are commercially also sold. Okay, when you just water it with the luciferin substrate to the roots, these plants will start glowing. Doesn't it look beautiful? Okay, so generally this particular gene is used along with your gene of interest. So whenever you want to screen whether the plant has a gene of interest, you just add luciferin to the plant, and if it glows, it means the plant got your gene of interest. Okay, coming to the next protein which is used as a reporter gene is green fluorescent protein. Okay, this is a highly many people use it, and this is the today every other vector which is used for plant and animal transformation have this particular protein, and you call very normally they are called as GFP. They call this protein as GFP or green fluorescent protein. So this particular gene is got from the jellyfish Aquaria victoria. So this is a jellyfish gene. So GFP is got from a jellyfish gene, and this is used along with your target gene to be transformed into or taken into the host organism. So those host organisms which acquire your gene of interest will also get the GFP, and you need not always look into whether your gene of interest are present or not, because I'm going to talk about a very cumbersome, very long technique for looking into the gene of interest directly, because you have to do it also. No, just by looking into the nearby person, you cannot say the gene of interest is present. But as being very positively thinking, when the gene of reporter gene is there, the gene of interest is also there, because they are neighbors. They are not one and the same. The reporter gene is present near your gene of interest. So when the reporter gene is present, or it is showing the character of the presence of reporter gene, it means you take it for granted your gene of interest is also there. Okay, so people have taken this GFP and they have used it for transformation, for using it in the vectors along with your gene of interest to get a and they have used this as a screening methodology to look for the recombinant organisms. And to be very the scientists, they don't stay at what they have got. They'll be doing research more and more, and they people have mutated this GFP to get a variety of color. So green fluorescence, so this particular gene, they produce a green fluorescent color. When you just show UV light on the organism, they will glow. Okay, they glow green in color. They have made mutations in these particular protein to get yellow fluorescent, YFP, yellow fluorescent protein, red RFP, red fluorescent protein, things like that. So this is an example of the animals which are transformed with the vector which has GFP as the reporter gene. Can you see this is the mouse which has the, so it means this mouse, when you show UV on them, they are glowing. So what you do is they generate make transgenic mice by the methodology what Dr. Mahalakshmi was telling. Okay, so they just use adenoviral vector, so many vectors they use, and they transfer the genes. And so when they transfer the genes into the animal cells, not all the cells may develop a transgenic mouse. So only a few of them. If you do the experiment on 4 ovum, only 3 of them may get the gene and 1 may not get. So you cannot just operate the mouse and find out whether the gene had gone into it. You just can glow UV on them and find out which one had got your gene of interest. Okay, so if you put insulin gene on them, you cannot find out whether this you have to do a very long experiment to find out whether this is making insulin. Rather, you can just glow UV on it and you find, okay, yeah, this is glowing, this has got my gene of interest. Okay, and the right panel shows the mutated forms of GFP: orange fluorescence, yellow fluorescence, green fluorescence, and red fluorescence. So this is the tetris fish. The wild type is generally transparent, it doesn't have any color, but you can have so many other so these are the fishes which are transformed with the GFP, which may be red GFP, orange GFP, yellow GFP, and gray yellow FP and green fluorescent protein. Okay, right. You have any questions about these reporter genes? Let me get into another important reporter gene. This is which is used for plant transformations. So, this is a cousin of what we saw in blue white selection, galactouronidase. Okay, so the same thing. This is another enzyme, a gene coding for another enzyme called as glucuronidase enzyme, and this is coded by the gene called as the Gus gene. It is coded by the Gus gene. So this particular gene is always incorporated into the vector when you do a plant transformation or when you transfer genes into the plant. So those plants which have got the gene of interest along with the marker gene will stain blue in color with when you add the X-gluc substrate is added to the media. So when you add X-gluc, the plants will turn, when you just add the X-gluc to the plant, they will turn blue in color when they have got the Gus gene. So the Gus gene is also used as a selectable marker. Okay. Am I clear? Shall I have a few questions for you people? Shall I have a few questions for you people?
Ma'am, is it possible?
Yeah, tell me.
Ma'am, is it possible for a recombinant organism to acquire only a reporter gene or a gene of interest that it's not get ligated together and inherited by it?
Yeah, you can do it. So what you're asking a very nice, beautiful question. You can do it. It means that these two may not be present in the same vector. They can be present in 2 different vectors. The gene may be used for just for the initial screening, and later they can be removed from the recombinant organism. That's what you asked, right?
Yes, ma'am. Ma'am, what happens when we apply in fluorescence technique and find glowing plants and animals, but it is possible that the gene of interest is not present in this?
That is why. Yeah, it means I told you it is your neighbor. This is not as such your gene of interest. So this is the neighboring gene. This is present near your gene of interest, and you take it for granted that when these genes are there, your gene of interest is also there, right? So when your gene is there, your gene of interest is also there. It's a subjective question. So you have to confirm the presence of the gene fragment what you have put inside to be present inside the organism, right? This is what you asked.
Yes, ma'am.
Okay, so now I'm going to tell you. So now your question will be answered in the forthcoming slides. So it is not enough that just you go for this kind of screening techniques where you can just by believing that the markers are there, your gene of interest is there. So it is subjective. It may be sometimes it may happen that markers may be there, your gene of interest may not be there. So there such kind of things may also happen. So that is why you go directly for a very high throughput and a very confirmative methodology for screening of recombinants called as the hybridization techniques. So right there, you are going to find out whether that particular fragment of DNA which you have transferred to the organism is present or not. Okay, so they are used to detect the particular sequence, whether it is you can use this technology for detecting the presence of a DNA fragment, an RNA fragment, or sometimes even or proteins are present, or target protein is present. So these methodologies are called as hybridization techniques. Okay, here they use the phenomena that DNA has a complementarity. Can anyone say what is DNA complementarity? Can any of you say what is DNA complementarity? Chat box. Students, please respond.
Then base pairing.
Base pairing, right? Can anybody say like how base pairing happens?
With a purine and a pyrimidine with hydrogen bonding.
Very good. So A pairs with T. So then the basics of DNA is A pairs with T and G pairs with C. So that is why the DNA is a double helical
AT
form.
and GC.
AT and GC, right? AT and GC. So this complementarity is used for these hybridization techniques. So generally, the DNA is present in a double stranded manner, and you in these techniques you just break the DNA into a single stranded molecule, and then you find out with what is called as a small single stranded DNA, which is labeled to find out whether your gene of interest is present. I'll just explain it in the in this. So here for this technique, what are the things basically you need is first you should have the organism which you want to test. For example, you take a plant tissue or an animal tissue, what you think as a recombinant molecule. You think that it is a recombinant molecule which has your gene of interest present inside it. And then you are looking for the presence of it, and to look whether it is present, you use a small piece of single stranded DNA molecule to go and detect the presence of the target gene. Okay, so this small fragment is called as a probe. It is called as a probe. Okay, so let me come into the so when you look into the hybridization techniques, there are three basic hybridization techniques to detect the different molecules. For example, southern hybridization is used to detect DNA fragments. So you can find out the presence of a DNA in a sample of a small fragment of DNA in a very large DNA molecule. And northern hybridization is used to detect a RNA to presence of a particular fragment of RNA, and western hybridization is used to detect a presence of protein. Okay, so the probe, as I told, the small single stranded molecule which is used to detect the particular fragment of DNA is for southern it is a labeled DNA, for northern it is a labeled DNA, but for western it is a labeled antibody. Okay, I'm going to now discuss about one particular technique which can extrapolate to the other two techniques, the southern blotting technology. So southern blotting it's a technique which allows the detection of a specific DNA sequence in a large complex sample of DNA. As one of the participants asked, ma'am, whether if the reporter gene is present, is it very sure that our gene of interest is present? It's not sure. You have to look into that particular target DNA if it is present or not. If we have such beautiful lens in our hands, it will be very nice. We need not go for such complicated procedures. We just have to go see with the lens. That cannot be done. No, that is why you are going to use a particular technology called a southern blotting technology to find out this. For example, if your gene of interest is present there, your human genome is very big. It is about millions and millions of base pairs there. If you introduce a small fragment of DNA, which is about 1,000 base pairs, and I want to find out whether the DNA is present inside the cell, you go for this technique. So this technique was first devised by Edwin Southern in 1975. So this southern blotting, it combines agarose gel electrophoresis for size separation of DNA and then hybridization. First, I will tell you like what should be done. So first, you have to take the whole. What we are now looking for is from a very large, very big, long strand of DNA. We are going to find out a very long, complex DNA. We are going to find out a very small piece of DNA which is of your interest. So for this, first you have to break the, first you have to take the DNA out of the cell. So you will be given transgenic. If I have a transgenic plant and I want to find out whether, for example, you take a BT brinjal, they will ask me to find out whether they have got the BT gene. I do a southern blot analysis. How do I do it? First, I take the brinjal plant and I extract the DNA. You have protocol to take out the DNA from the brinjal plant. You take the DNA from the brinjal plant. Okay, you take the DNA from the brinjal plant, and then you have to first do you have to separate the DNA. First, you break the DNA into smaller pieces using restriction enzymes. So I told you yesterday, Dr. Singaravelan told you about restriction enzymes. No, they can cut the DNA at specific spots. So you just randomly cut the DNA into small pieces using restriction enzymes, and you just make them to run on what is called as an agarose gel. Agarose gel is nothing but a jelly-like medium or matrix. You can call it as a matrix. You can just put the DNA on these matrix and you apply electric current. The DNA will start moving, and the movement will be such that the smaller DNA molecules will run faster, and the larger DNA molecules will be lagging beyond. Okay, so you can see when the DNA is you just run the DNA and the agarose gel, and then what you do is so the gel, whatever I'm talking about, the agarose gel is a jelly-like thing. So you cannot go for detection of the DNA on the gel because the gel may break. It is very flimsy, like jelly. You cannot hold it in your hand; it will break. But the DNA can run only on a gel. So first you run the DNA on this gel. That is, you take the DNA and you allow it to size separate on this gel. And then after you run the DNA on the gel, you transfer the gel or the DNA, or you take the DNA into a more sturdy substrate. You have to transfer the DNA from the more flimsy or breakable substrate into a more sturdy or unbreakable substrate. So whatever substrate we are here, what you use for southern blot, they are membranes. Okay, so you call them as membranes. Okay, and then you take it into the membrane. So I'll tell you. So this is a step one, how DNA is separated. First, you take the DNA, and the DNA is broken with restriction enzymes, and you can see. So these are broken into smaller pieces, and then this is an agarose gel, gel, where you run the DNA. So you run the DNA. Okay, then okay. After you run, this gel is jelly-like. Okay, after you run the gel, you take the gel and you transfer the DNA from the gel into a sturdy substrate and matrix. They may be membranes. So the membranes are very tough. They are unbreakable. They are very flexible. And such scientists have made nylon membranes. Okay, they will be like paper, but they will not be torn. And the DNA can be from the gel. The DNA is transferred onto the membrane. Okay, now your DNA is taken into the membrane. Okay, so before taking the DNA, so DNA is double stranded or single stranded?
Double strand.
Double
Double-stranded molecule. So before you transfer, you call this methodology of taking the DNA from the agarose gel to the membrane is called transferring. So before you transfer the DNA into the membrane, you make the DNA single-stranded. That means that You break the DNA, the hydrogen bonds. When you break the hydrogen bonds, the DNA will be made single stranded. So there will be single stranded DNA. So in the how do you do is you just take the gel and you soak it in sodium hydroxide. So alkali. So when you put DNA in alkaline condition, the DNA will break and they will make a single stranded molecule. Okay, so this single stranded molecule, which is on the agarose gel, you transfer them onto a very sturdy membrane like nylon membrane. So the methodology is like this. So you just take a bowl or a very shallow bowl. Okay, in that you fill it with buffer. So on the buffer, you put a small platform, and on the platform, you just put some what is called as a wick. You know what is a wick? Wick. A wick. What you use? A wick. WICK. So when you put it in lamps, no, it's called. So you wicks means they will absorb water or oil, whatever. When you put it in the lamp, it will absorb the oil, and you take. So it is called as a wick, right? Tamil you call it as thiri. So similar manner, here you use a paper which serves as a wick, which will transfer the buffer, or it will the buffer will raise onto the this particular paper. So on that you place the agarose gel. You place the agarose gel which carries the DNA. Okay. On that agarose gel, how do you the transfer of the DNA into the nylon membrane? That is what the protocol I'm describing. On the agarose gel, you put the nylon membrane. Okay, on the nylon membrane, you stack a stack or a row of column of filter papers. You put a column of filter papers, and on that you put some weight, so that the weight should have press. Okay, press the setup. So this is the setup. So in this setup, what will happen is the buffer in the tray will start moving up. So because you have a wick here, you know, like whenever you put you just put a small thread or something inside our water, so the liquid will start raising into the setup, and as the buffer is raising, it will get into the gel, and from the gel it will go into the filter papers. It will be soaking. The papers will be soaked with the buffer. So while the buffer is traveling from the bottom to the upper part, so on the way you can find the gel and the membrane touching each other. So while the buffer is moving, it not only will move alone, it will also carry the DNA from the agarose gel and it will place it on the membrane. So because of this is called as capillary action. Because of the capillary action of the wick and the whatever the filter papers, the buffer will raise up to upwards, and while it is moving upwards, it will take the DNA from the agarose gel and put it on the membrane. So, so you leave the setup for about 12 to 16 hours, and you see that after 16 hours, the membrane will carry the single stranded DNA. Am I right? So the agarose gel, the whatever the DNA which was present in the agarose gel will now be transferred into the membrane. Now in your hand, you have the membrane which can be very easily handled. It will not be as flimsy as the gel. Gel will break like jelly. But your membrane will be there very sturdy, and you can do whatever experiments you want. Now you use this membrane to probe. On that membrane, you have the DNA, whole DNA. If I take this whole cellular DNA, in which your DNA of interest will also be present. Now on that, you are going to find out where whether your DNA is present or not. For that, you're going to use what is called as a probe. So this is a very diagrammatic representation of the membrane which carries your DNA. Can you see this picture?
Yes, ma'am.
Yeah, so this will have a single stranded DNA which will have the protruding hydrogen bonds present. Okay, so this puppy, so it is a very long, it's a pictorial because it is showing very short, and you can see that among this DNA somewhere you will have your target DNA also. Okay, for that, to detect this, you have to prepare a small DNA molecule, and this DNA molecule will have the complementarity to your target DNA. It should be complement to your target DNA. If your, for example, if your target DNA carries a sequence of A, T, G, C, this probe should have a T, CAG. So it should be complementary to your target sequence. The probe can be about 500 base pairs, and the probe will also be a single stranded molecules. So when you add this probe to this membrane, where will it go and bind? Where will it go and bind? If you call it a probe something to find out, where will it go and bind? Will it bind to all the pieces of the single stranded DNA on the membrane? So you have so many fragments of DNA. Will it bind to all the fragments? Huh?
It will bind to the complementary one.
And complementary one is which is the complementary
Gene of
one?
interest.
Yeah, it is your gene of interest. So it will go and bind to those areas where your gene of interest is there. So it will only bind to those areas where your gene of interest is bound. So, but what is the meaning? If it goes and binds, how will you find out whether it has bound or not? Okay, so you don't know. Okay, you do the experiment on the membrane and you put you take the DNA in a solution, you put it on the membrane, and you don't know whether it has bound or not. So for that, what you do is you put a label on the probe. You put a label on the probe. Okay, so this label can be of it's nothing but a radioactive anything. It can be a coloring molecule, or it can be a radioactive molecule. Now I'll talk about a radioactive molecule. You put a particular, so in this probe you incorporate radioactivity in it. You incorporate radioactivity. So radioactivity, it means it will be emitting high energy radiations. Okay, so it will be emitting high energy radiations. So it means this probe, which is labeled with radioactivity, when it goes and binds to the membrane on the areas when it goes and binds to the membrane on the areas where there is your target DNA, those will become radioactive, right? So those areas where the probe binds is your target gene. Okay, so the target gene wherever the probe had bound will become radioactive, right? Can you see this picture? So the probe binding area will become radioactive because the probe is carrying a radioactivity. Okay, so now we have a methodology to find. So if you can find out where there is radioactivity, then if there is radioactivity, it means what it means. If on the membrane, if there is radioactivity, what does it mean? If there is students, please tell, am I audible? Only when you tell, I believe you.
That it contains the gene of interest.
Okay, if the probe binds to any of the places on the very good effort, so it means that whenever the probe binds, it means that it is your target gene is there. If it is not binding, it means your target gene is not there. Okay, so presence of this probe or presence of radioactivity can be deduced by a method called autoradiography. Okay, so after first you do run the DNA, you transfer the DNA into the membrane, and then you use the probe to hybridize. You call this is what is called hybridization. Hybridize. So this probe goes and binds to the complementary sequence. This is called as hybridization. Okay, so wherever the probe had bound, it means your target DNA is present there. Okay, now the membrane will show radioactivity. So it may if only when your gene of interest is there. When your gene of interest is not there, it will not show any radioactivity. Now I'll show you how to measure the radioactivity or visualize the radioactivity. There is a methodology called autoradiogram. So autoradiogram is nothing but using a X-ray film. Have you seen an X-ray film? Girls, have you seen an X-ray? Students, have you seen an X-ray film? So X-ray film, when you go for an X-ray of a broken leg, they will expose your leg to, they'll put you in a dark place, and they will just flash X-rays on it, and then they will take that, they will just wash it, and they will show you. So wherever there was your bone, you will have you will have a very clear place, and other places will be dark. No, so similar manner, this is so you use the same X-ray sheets. You get the X-ray sheets from the Photoshops, and you place it on the membrane. So you do it all in the dark. you cannot expose the X-rays to light. You put the X-rays on the membrane and you expose the membrane to the X-rays for a few minutes. So what will happen is few minutes or hours. So what will happen is the radioactivity, on the membrane, so for example, there are two fragments. Fragment A is your gene of interest and fragment B is not your gene of interest. So your probe will bind only to the gene of interest and you can see radioactivity only in this place of the membrane. So you place the X-ray on to the membrane, right? The size of the membrane, and keep it in a dark place for few hours. After taking this, you just develop the X-rays. So when you develop the X-rays, you can find that, so wherever there was a radioactivity, this radioactivity, that is, they will be emitting radiations. This radioactive radiations will go hit the X-rays and will make a dark precipitate on the places where the radiation falls, because of which that part of the film will become very dark in color. So yeah, so and then you have to wash, like what you do, you can see in movies, no, in all horror movies, where they will be washing it in a red color room. You call it as a dark room. You just wash it with wash it and the fixers and developers, and then you take out, and then you can see that wherever there was a radioactivity, you will have a very dark spot on the X-rays, which shows that that particular piece of DNA has your gene of interest. So this is my, actually this is a real-time experiment. This is my PhD work. So this is the agarose gel, where I have run the DNA and the stained with ethidium bromide, and this is called as an autoradiogram. This is the X-ray which was exposed to radioactivity. Can you find these dark spots are the places where? So this is the DNA, and the corresponding to those DNAs, you can find the dark spots. What are the dark spots? What are the dark spots?
Gene of interest.
Gene of interest.
So for example, this is one sample of DNA. Okay, so you can take vertical columns. This column is one DNA, and then next column is another DNA. Sorry. And the other column is another DNA. So you can see that. First sample has the gene of interest. Second has the gene of interest. Third one, does it have?
No, ma'am.
Okay. The first one, how many copies of the gene of interest they have? How many genes of interest is there? How many fragments do you see? Upper one and lower one. How many fragments? 2 fragments are seen. That means 2 genes of interest are present in this particular sample. Okay. And similarly here also. In the 5th sample, you have 2. So this is how. So this is a hybridization technique, and you call it a southern hybridization, which is used to find out the specific fragment of DNA. Okay. So similar manner, you use when you take this. So this is a DNA sample which I ran an agarose gel. When you run a RNA sample and you do the same technique, RNAs. Okay, you know, like mRNAs, and you take the cell, open the cell, you can extract the mRNA and you run it on the gel. And when you look into it, you will find. So you do the same experiment. When you find the signal, it means you are looking for the presence of a particular RNA, and that particular technique is called as northern blot technique. So first, because southern was the first one which was established, this particular technique was given the name of the inventor. That is the person who devised this technique as southern, and northern is a small variation from southern, that is using the RNA. And because it was called as DNA was called as southern, the RNA analysis was called as northern. So similar manner, you can also take proteins. You run it on the gel, and you also screen for a particular protein. You call it as westerns. And for western, you cannot use a DNA as the probe. You have to use an antibody as a probe. You have to use an antibody as a probe. Okay. I think with this, I will finish my talk, because I don't want to talk more. I think I wanted to talk about PCR, but I'm running out of time. Okay. Thank you, students.
Thank you, ma'am. Students, if you have any questions, you can just post it in the chat box. Now the session is open for questions. You can just post it. If you have any questions, participants? I think ma'am has given a very clear explanation. I think everybody is very clear with what she told. I think there is one question.
Ma'am.
Does YFP, RFP can be used for phage vectors?
Generally, YFP and RFP are not used in phage vectors. These markers are used only for eukaryotic organisms. Okay, phage vectors they only use for cellular, so it's not used for those things. It's not used in phage vectors.
Next question. Does the probe cause any disformalities?
Actually, you do this experiment in vitro. You do not do it inside an organism. You just extract the DNA from the cells, like what Mahamam told. Whenever you do an experiment outside the cell, you call it as in vitro. So there is no question of any making disformalities.
Any more questions, participants?
Which type of human growth hormone?
Which type of human hormone was used in super mouse?
Human growth hormone 2 was used for making the super mouse. Same hormone has been used in goat, sheep, and all those things. Fish, to make a super fish.
Does the labeling inherited from one generation to another?
I don't understand the question. Labeling in the sense recombinant DNA. If you're asking about the recombinant DNA, which is transferred into the living organism, if you're talking about that, yes, for sure they are inherited. It has been checked, and it has so there is a stable inheritance of the genes because they get into your DNA, and when the DNA replicates, those genes are also replicate. But when you talk about labeling of the probes, as I told, probing is done only outside your body, so there is no point of inheritance there.
Participants, if you have no more questions, then I think we can wind up this question session. Now I invite Dr. A. Lakshmi, Assistant Professor of Chemistry, to propose the vote of thanks.
Gratitude is not in the words, but in the heart which expresses it. A very warm afternoon to one and all connected here. On behalf of the organizing team, it's my pleasant privilege to propose the vote of thanks on this occasion. First of all, I thank Lord Almighty for showering His blessings to complete this session in a fruitful manner. My heart fills with lots of gratitude for our resource person, Dr. R. Sripriya, for sparing her precious time in enriching our young minds. I am sure, ma'am, students have acquired more knowledge about GMO, creating a GMO, and various screening methodologies. You made the session very clear and more interactive. Thank you, ma'am. I thank our secretary, Reverend Sister Flora Mary, our principal, Reverend Dr. Sister A.S.J. Lucia Rose, Deputy Principal, Reverend Dr. Sister Kalandathras, for their support, vision, and commitment. I take this opportunity to thank Dr. Sister Arokya Genetius Alphonse, the overall coordinator for DBT Star College Scheme, and Dr. Sister Jessie Fernando, Head and Associate Professor of Physics, for their valuable contribution, guidance, and encouragement in all our efforts. I owe a special gratitude to all the heads and organizing team members for their untiring effort for the success of this event. I owe a special gratitude to the technical team for their continued support. Last but not the least, I thank all the participants for their enthusiastic participation. Thank you, one and all.
Thank you, ma'am. Thank you for the organizers.
Thank you, participants. I have a few instructions. Now the feedback link will be posted in the chat box. Do fill it. And attendance is mandatory. And this feedback link will be posted only here in this Zoom meet. It will not be posted further in any WhatsApp group and all. So attendance is mandatory for the issue of the e-certificates. And evening, the quiz will be posted in the Google Classroom. So kindly fill in the feedback link, students. I think it's posted now in the chat box. Thank you, students, for your active participation. Thank you. Tomorrow, we will meet at 10 o'clock.
Ma'am, link is not there.
Students, kindly wait. I'm posting it. Just wait.